Welcome to the BioWhisperer channel. Our topic today is on a perspective overview of microbial immunological investigation, which is part of medical biotechnology coded under Red Biotechnology. Let's begin with an introduction to the term, immunological techniques, from Nature Portfolio. Broadly, it is split into two parts. First, the experimental methods to study the immune system. Second, it encompasses the approaches, strategies or research methods to generate or use immunological reagents as experimental tools. Within the study of microbiology, the most common immunological methods relate to the production and use of antibodies to detect specific proteins in biological samples. Currently under the immunological diagnostic techniques or approaches we have the following. First, we have enzyme-linked immunosorbent assays, Western detection, the immunofluorescence assay and the hemagglutination inhibition assay when dealing with human clinical samples. Let us quickly review the culturability factors and concerns. Some microbes are unculturable within lab environment, meaning we can only collect them from their normal habitat within the environment such as a soil or water sample. Another study on bacterial within our oral cavity where only a small subset are culturable. Let's take a look at an example. The sexually transmitted disease, syphilis, is caused by the spirochete Treponema pallidum, which remains unculturable today. Some pictorials from Google image search are shown on the screen. I would thank you for helping to share your love for this science channel by clicking the like button and subscribe for future updates. With that out of the way, let's continue the discussion. The viable but non-culturable, also termed as VBNC, state is a unique survival strategy of many bacteria in the environment in response to adverse environmental conditions. VBNC bacteria cannot be cultured on routine microbiological media, but they remain viable and retain virulence. Other factors to consider when attempting to culture microbes within the lab includes practicality, which encompasses the ease of culturing and if the strains are highly pathogenic and infectious. The availability of appropriate biological safety certified labs is also a factor. Finally, as we have shared earlier, some microbes are just unculturable, or at least we have not yet figured out how to best culture them. Omics investigation is one potential investigative approach. Back to the issue of unculturability of bacteria. The lack of key nutrient or toxicity of culturing conditions are areas to be addressed. The disruption of bacterial cytokine networks, where bacterial cytokines are thought to be the mediators of bacterial to bacterial signaling, will affect the growth of bacteria within bacterial communities such as biofilm formation processes. The separation of bacteria during lab culturing on solid media would disrupt such networks and may explain why some organisms are unculturable. So why should we adopt an immunological detection approach? For molecular identification of microbes, scientists often make use of biological macromolecules such as proteins and nucleic acid sequences for elucidating the identity of the microbes. Nucleic acid based molecular detection techniques have revolutionized diagnostic virology with their faster, highly sensitive, and highly specific diagnosis, and is useful for detecting COVID virus variants in the current pandemic. Proteins being the expressed gene products and are easily accessible on the microbe surface as antigens present a good diagnostic opportunity for researchers. Immunological responses mounted against the microbial antigens such as antibodies can be detected as well. This is part of the serological investigation. During an active infection, doctors can detect neutralizing antibodies after active immune response is mounted. Antibodies recognize the antigens and bind to them. Serology works on the basis of antigen-antibody recognition. Our immune system has a specialized search and destroy army. Antibodies are specialized, Y-shaped proteins that bind like a lock and key to the body's foreign invaders. Within the antibody lies the antigen binding sites, which is constituted by the variable regions of the light and heavy chain forming complementarity determining regions. Disulfide bonds connect the antibody chains together and they can be fragmented into three parts, namely the FV, the FAB and the FC fragments. The enzyme papain can break off the antibody into two fragments, FAB and the FC regions. IgG structure is the most well-known type of immunoglobulin because it is the most abundantly expressed and secreted by our organism. IgM is the first isotype to be expressed during B-cell development. After activation and switching, B-cell ceases to express IgM and IgD and starts expressing IgG, IgE or IgA. IgA is the main isotype found in secretions like saliva, tears, and milk. IgE antibodies can be found at very low levels and exclusively beneath the skin, mucosa and in the connective tissues of blood vessels. Detection of IgM and IgG are of diagnostic significance. IgM levels peak are indicative of a recent active infection, whereas past or chronic infection or vaccination give rise to IgG, which are also sometimes more commonly termed as neutralizing antibodies. The road ahead. It's about making progress in culturability. One of the challenging aspects is mapping the human intestinal microbiota, which comprises perhaps hundreds of bacterial species. Most of them are regarded as unculturable and have never been isolated in the laboratory. The authors of this Nature paper performed a targeted phenotypic culturing linked to large-scale whole genome sequencing, phylogenetic analysis and computational modeling, to cultivate a substantial proportion of the human gut microbiota. This showcases the integration of omics approaches to help with the understanding of complex environments, and this paper uses the gut microbiome as an example. Thank you for watching this video. If you like this video, please smash the like button and subscribe to this channel for future updates.